Good evening and welcome to the 2020 Shaw Prize Special. 3 European scientists have won the Life Science and Medicine Award for the development of optogenetics, a revolutionary technique that's allowing neuroscientists to trace and control brain activity with light. Thousands of labs around the world are using the technique to unravel the complexities of the human brain and even develop treatments for brain diseases and mental health problems.
Controlling minds with light. What sounds like science fiction has been brought to reality by neuroscientist Gero Miesenbuck. The Austrian scientist arrived at the University of Oxford in 2007. Here, his team can put flies to sleep and make headless flies take flight using nothing but light. Application of the optogenetics technique to manipulate an organism's neural circuits shines a light on how neurons drive behavior and intelligence.
I think that discovery is not just that you find an objective fact that's somewhere out there in the universe and you just record it like a passive observer. There's an element of creation also in discovering how nature works. And you conceptualize how you think a certain process actually runs. And that creative element is, I think, common to, for example, a writer, a novelist penning down a novel. I think it's very, very important not to suppress this artistic element in research.
Despite pioneering a whole new field of neuroscience, Miesenbuck's childhood dream was not to be a scientist at all.
My dad was a classicist, and at some point I thought I might follow him into literature. But he advised me very strongly not to do that because, he said, the potential for discovery in the sciences is so much greater. You have an essentially unbounded universe of things that you can figure out. So I took his advice and became a scientist, but I've retained an interest in literature and also in writing. I think some critics might say that I'm still writing fiction when I write my scientific papers, but that's of course not true.
The 55-year-old gave up literature for medicine in university. However, born with the desire to create, he soon realized the repetitive work of a typical doctor was not for him. When he was finishing his medical training, he came across an unusual research paper, or what he called artistically presented scientific literature. He wrote to the author, James Rothman, and asked to join his lab at the Memorial Sloan Kettering Cancer Center in New York.
So my postdoc advisor, a fabulous scientist named James Rothman, who won the Nobel Prize in 2013, he was probably the dominant influence in my scientific upbringing, because he showed me that there is something like a personal style in science. One of the things that attracted me to the particular lab that I joined was that the scientific research was framed and presented in this artistic, aesthetically appealing fashion. That really attracted me. It was not just, as is sometimes dismissively said, stamp collecting, where people just observe and collect various observations. It was all driven by ideas rather than facts. The idea to optogenetic control came to me one Saturday afternoon while I was not really thinking about the problem, but I must have been sort of incubating it, and it suddenly sort of popped to the conscious surface like a bubble that reached the surface of a pond of water. I realized then and there that this could have a profound impact if it was feasible. So the next difficulty then, of course, was to try and find a genetically encoded receiver that could take a light signal and transduce it into electrical activity. And then it also became logically clear where one could find such genetically encoded receivers. And that is, of course, among the light receptors in our eyes and in the eyes of other animals. So the idea then was to take one of these light receptors and transplant it genetically from a light-sensing cell in the eye to a different cell deep in the brain. And if illumination of that cell deep in the brain then caused electrical activity, just like illumination does in the eye, then the approach would succeed.
The technique, when applied to specific neural circuits in the brain, would enable scientists to find out in unprecedented detail how each cell type contributes to our thoughts, emotions, and behavior. It took Mysenbach about 2 years to bring his idea to reality. This video captured the first use of optogenetics to control animal behavior. Cells in the fruit fly's brain were engineered. Light caused it to take flight in the petri dish.
When I hear about such techniques, I think that the person behind who had the idea to put such a technique in place must be a very creative scientist that has such a long-term vision that's going to help to change the field and the way we think about science. I would describe him as a very visionary scientist who's got big ideas and always thinks one step further than probably most other scientists would do. So I feel it's a great honor to have the opportunity and the possibility to work in his lab to get inspired by this vision that really pushes us to go further and further and further in order to address very ambitious questions and in order to be able to change science.
I think optogenetics is really revolutionary because so before neuroscience was just more of a field of passive observation of neuroactivity of the brain, but now with optogenetics we can actually actively engage or actively interrogate the nervous system and add our own tailor-made activity into the nervous system and see the consequence to other neurons or to the organism's behavior in general. And I think that's why it's so revolutionary, and yeah, that's why it transformed neuroscience.
But Mysenbach's ambition doesn't stop here. He continues his research with fruit flies.
I think the technique of optogenetics has matured considerable over the more than 20 years that it has existed now. There are, of course, still areas for improvement, but I have to say I'm not working on these issues anymore. I invented optogenetics because I knew I needed a technology like this to solve some of the neurobiological questions that interested me. One of our main interests at the moment is the neuronal regulation and the biological function of sleep. And optogenetics was extremely important in pointing us to the areas in the brain that are important for sleep and for wakefulness. We've recently uncovered a surprising link between energy metabolism and sleep that puts a physical basis to the long-standing but previously very difficult to test idea that energy metabolism, oxidative stress, lifespan, aging, neurodegeneration and sleep are all somehow connected. I think we may have found sort of the linchpin that connects all these different things.
Over the years, scientists around the world have continued to improve on the technique. After the break, we'll look at the collaboration between German scientists Peter Hegemann and Georg Nagel. The pair together discovered simpler light-sensing proteins from a type of green algae, faster and genetically simpler than the fruit fly genes used by Mysenbach. As Austrian scientist Gero Mysenbach first succeeded in activating nerve cells with light, a German team made a groundbreaking discovery that propelled the field of optogenetics even further. Peter Hegemann of the Humboldt University of Berlin and Georg Nagel of the University of Würzburg. Channel Rhodopsin.
There are two words it is made of, opto, which has, of course, something to do with vision or with light, actually more with light, and genetics, because there's a gene transfer. So it's always protein you take from one organism, mostly actually from microbes, bacteria, or little algae, and then bring it into an animal, for example. It became very simple since we discovered the generodopsin.
Now about 1,000 or 1,500 labs in the world are using generodopsin from chlamydomonas for their experiments. And I think the biggest advantage is the ease to use it. It's just one small protein which unifies a channel and a sensor. And the next thing is it's robust.
The team extracted this wonder protein from a simple green algae, Chlamydomonas reinhardti, dubbed by scientists as the green yeast for its powerful role in dissecting biological processes. It all started in 1985. Fascinated by how the unicellular organism moved towards and away from light sources, Hagemann began to study its light perception. Back then, he did not think his attempt to understand this natural phenomenon would lead to applications in optogenetics.
We would like to understand how algae behave, and this is what we use photoreceptors for behaving and orientation in our light environment. And animals do it, and algae do it. They can move and orient to places that are optimal for photosynthetic survival and photosynthetic growth. And this is per se an interesting question. So application is the second part of the research. If we don't search for novel natural phenomena, we have nothing to apply.
This mystery took him over 15 years to solve, and the real breakthrough came when he joined forces with Georg Nagel.
It took a few years. I mean, in 2002 we published the first study on generodopsin 1, but this protein was not expressed very well. In 2003, with generodopsin 2, everything changed. It was clearly useful. And then several groups used it, and in 2005 then several papers came out from different groups, all referring to our study, and also they have gotten the DNA, the genetic material they got from us, and could show that it works beautifully. And then the growth was somehow exponential.
After we have proposed that the photoreceptor in Chlamydomonas is a light-gated or a light-activated ion channel, the people didn't believe us. So to demonstrate this, you have to take this single protein and express it in some host cells to demonstrate that this is indeed a light-activated channel. And that important step was achieved with Georg.
The science world has a long history of trying to stimulate nerve cells. Back in the 1700s, Italian physician Luigi Galvani used an electrode to pass an electrical current into frog nerves. The action caused the frog leg to twitch, from which Galvani concluded that neurons could use electrical signals to pass information. Modern brain surgery pioneer Wilder Penfield started stimulating the human brain with electrodes in the 30s. Electricity was applied to certain areas of a patient's brain to understand the parts of the body it controls, which allowed him to map the human brain. With the development of optogenetics, cell control can be much more precise, non-invasively, with light. Not only do neuroscientists apply the technique to understand how the billions of neurons function, there are also studies aiming to look for treatment of disease.
There are experiments, I mean, therapeutic essays underway, even with blind patients, where they want to bring in generodopsin into the eye And I think there's a good possibility that this even might help and restore vision in blind people. At least it was shown for mice. And it is probably much better instead of inserting some electrodes or some chips into the eye with lots of electrodes or so. In principle, it should be much more harmless.
You can also apply it for Parkinson's diseases or epilepsy and many other diseases, but it's not so far developed. And the work on application is complicated because you have to introduce the encoding DNA into the cell of interest. And if you want to do it in humans, as everyone knows, this is not free of ethic considerations that we have to overcome or we have to justify before we go into a human cell.
The two teams are now testing new proteins so they can continue to sharpen these techniques.
We want to bring in different tools. This is not only generodopsin, these are also other light-sensitive proteins which either come directly from microorganisms, bacterium or an alga, or which were modified by us and are, of course, light sensitive. I think it's always ideal if they are only sensitive to blue light, so you can still watch your organism in red light because you are not activating the protein. And then with the blue light, you can activate this protein and see what is then the outcome of your activation, of your manipulation.
What we are working on at the moment is to shift the spectrum further to the infrared, because infrared light would be more ideal than visual light, because the penetration in the brain is much higher. Then we combine different tools, and we like to bring into the business light-activated enzymes, light-activated cyclases. This is a gradual improvement. If you want to jump onto a new level, you probably have to change the media. Try to find proteins that are sensitive to ultrasound or proteins that are sensitive to radio waves. If you achieve this, then you have reached a different level, a completely different level. But we need young scientists, maybe coming from different fields.
Inspirations they bring to the field of optogenetics go beyond their discoveries.
He's a very, very nice person. He's an excellent scientist, and I think everybody who is in touch with him feels this. And he has a very, very, on the one hand, relaxed attitude, but also a very deep understanding of science. So when you discuss whatever problem with him, it is always very, very productive and illuminating.
Working with Peter is a pleasure, I would say, because he is an ambitious scientist. He's curious. He has a lot of fantasy, and he never stops halfway, I would say. If he's tackling a problem, he wants to solve this problem, and he wants to bring the solution to an end. He also listening to other people's opinions in a critical way, always open discussions, and that's what makes these cooperations so fruitful.
What I've learned from interacting with Peter, or through understanding how he has decided to run his scientific career, for me this is kind of like a role model situation. Sometimes it's worth it to not just trying to maximize your impact and do everything as fast as possible, but also just figure out what is really interesting for you, and maybe also just take the time to really get to the bottom of it, because sometimes you don't know it, but it will really be worth it. Say again, in the case of the use for neuroscience.
Looking back at solving these problems looks easy now, but there were many obstacles along the way. These pioneering optogeneticists summarized their success with a single word: persistence.
After discouraging time period, when you work on something for a long time, you might get frustrated, and you might take your own perspective or life into question, but if you're a real scientist, it comes back quickly. You think about different alternatives and new approaches, and then the enthusiasm might come back quickly. But it goes up and down. It's in every job, probably.
I think I felt more tired when I was young because it takes so long, and sometimes I thought, oh, I would rather be a bus driver than you know what you are doing, and I didn't see the outcome. And I must say I was very lucky in my later years. I guess the most important thing is that you enjoy what you are doing and that you find projects and experiments which you like to do, where you want to see what is really happening in nature, and then even if you have to work for longer times or so, then it's not so bad, and it gives you something back.
That's our show for this evening. Next time on Pearl Magazine, we take a look at how those living below the poverty line stay afloat amid the COVID-19 pandemic. Please join us then. Have a great evening. Bye for now.